Thank you for that introduction. Yeah, so I'm really excited to be here and tell you more about DataPLANT and what we are doing in order to develop and provide tools and services for structuring the data jungle. So in sort of classical publication timelines, generally scientists acquire data in order to discover knowledge. So usually a large set of data is acquired or collected, and then only a subset of that or selected data is then translated into knowledge in the form of research articles, publications, presentations, that sort of thing. But in science, all data is valuable. So whether or not it is published, there is still the potential to learn new things. So this data then can be reused, to regain further knowledge, or either in and of itself or else coupled with maybe newly acquired data. There's also always new techniques or theories that may require this data to be re-examined. So it's important that we don't lose this data. So even if it doesn't end up in original or in a journal publication at first, it may be useful down the line. So we believe that becoming fair will drive science. So increasing the level of annotation at the very source and tracking provenance over the course of the experiment or the course of the project using community standards will help to maximize both the findability as well as the reusability of data. I think this is something that is easier said than done for anyone who has had to maybe go back in a lab notebook or find a piece of data from 6 or 12 months earlier. This can sometimes be quite challenging. So there is a great deal of effort that scientists or researchers have to put in to make sure that their data is fair. So just a bit about DataPLANT. So we are part of the NFDI, or the National Research Data Management Infrastructure, and we're working to provide tools in order to structure research data according to the FAIR principles. So in this way, the researcher can effectively manage their own data, as well as annotate this data with the relevant metadata as early as possible. And in DataPLANT, we are focusing on the entire experimental timeline. So from the very beginnings or the conception of a research project or the beginnings of an experiment through to the collection of the data, the analysis of the data, and then finally uploading that data into repositories as well as the publication that may come from this as well. So really want to focus on all aspects of the research cycle and not just experimentation or publication or something else. So a central component of the DataPLANT management strategy is the ARC, or the Annotated Research Context. And what an ARC does is pack an entire investigation into sort of a single unified entity. So there are places for, brainstorming sessions about what experiments to do, all the way through to performing those experiments, maybe analyzing that data, developing the visualization needed for a journal publication, and then all along providing the ability to annotate this data. So provide the metadata or data about the data in order to really give a context for what the data actually represents. So I think there is a huge responsibility on researchers to understand and manage domain relevant standards. So what I mean by this is if you are performing an experiment, you need a particular piece of equipment to gain your measurement. There are going to be a series of checklists or vocabularies or even standards that are needed to run this piece of equipment. If you're working on a more complex experiment, you're going to end up with multiple different checklists or standards depending on the equipment that you're using. And then once this data is then collected, you have to analyze it. And there comes a whole nother level of different sort of workflows that require either certain parameters, certain standards, or use certain vocabularies in order to analyze the data. So this is already a huge responsibility for the researcher to keep these things straight in order to properly apply them to different parts of an experiment. On top of all this, once we get to the point of finally getting to upload data to the public and to the world, you have different requirements outlined by various repositories. So depending on if you're planning to submit to GEO or metabolites, they're going to require different things from you to describe your data. So what we're hoping to do with the ARC is to give a single entry point of logic for data management and computations. So pack all these different checklists and standards and vocabularies into one easy to follow and standardized structure, so that researchers can easily go back and refer back to certain vocabularies or standards that may be required. But in this way, nothing gets lost, and theoretically, everything has a place. So what is an ARC? So a real-life ARC is a user-friendly scaffold for research data and analysis. So basically, it's a folder structure, but it's structured in a way that's intuitive as well as standardized, and can be standardized across a whole series of different domains. So we also have a system for tracking changes as well as ensuring provenance, which I'll get to in a little bit. So we've built our ARCs on top of already existing standards. So the ISA model, so investigation, study, assay, as well as common workflow language, or CWL, are both incorporated into our ARCs, which means that using these already existing standards makes ARCs not only more intuitive, because researchers may already be familiar with these standards, but also easily transferred between different researchers. So here is an example of how data might be structured or organized within an ARC. So the top folder here are the studies. And if I'm doing, say, like a DNA extraction for DNA sequencing, I might put here sort of, where I got the seeds for my plant from, how the plants were grown, how they were harvested, and I can even go as far as, how these leaf samples were then processed. Maybe they were freeze-dried, and even to what rack of what minus 80-degree freezer these samples are now sitting in. So just easily organized and easily structured, so that you can always go back and find the exact sample that you need as quickly and efficiently as possible. Next, we have the assay folders, and this would contain information about how a sample went from being a sample through to a measurement. So this would contain information about the process of the DNA extraction. So the protocol that was used, the quality as well as the quantity of DNA that was extracted, what sequencing center the samples were sent to and when, and as well the raw data that then was received back from the sequencing center. So all this would then be located in the assay files. Next, we have our workflows, and this would outline or detail how the process of analyzing the data looks like. So what tools were used in the tool chain, what input files are, and what the then corresponding output files are. And in this way, you're able to sort of structure the process 1, 2, 3. This is how my data needs to be analyzed. In the runs folder, we have the specific instances of this. So I'm sure anyone who has had to work with multiple tools or analyze different subsets of data know, you use the same tools, but you might use them differently. You might adjust parameters or, use different input or output files. And so all of that information is contained in the run. So how you specifically ran your data using the workflows to get your final results. And then finally, we have the investigation folder, and this contains all the administrative metadata around the project. So it really sets the context for a researcher coming in who doesn't know much about the project. It outlines, the main goals, the main bits of information, the main people involved, that sort of thing. So you can really understand the context on which the rest of the ARC was built or developed. Another important characteristic of the ARC is that we are working to make things as flexible as possible. So many experiments, you might be doing the same thing with 500 samples. And this does happen at times, but I think any researcher knows that the second the target material, maybe you change your plant species, maybe you're looking at roots instead of leaves, things have to change. So that could mean a changing of protocol, that could mean combining different protocols in order to get the exact data or the best quality material or best quality sample out at the end of the process. And for that, we have developed this modular scheme. So you can easily add or remove characteristics or parameters. If you do have more steps in your experiment or more things to consider, you can easily swap out or remove or add these different components in order to make annotating as quickly and efficient as possible. So to explain a bit more about the tool that we use to do this, we came to where I think most researchers are comfortable. So probably even most people are comfortable in a just basic row and column based environment. So the spreadsheet. So things like Excel, where you can easily add in things in an organized structure, and you can therefore easily annotate your metadata without having to learn a completely new system. So for the example here, we have our source, and then you can select the series of parameters. These are then, we consider our parent terms, and then our tool provides a list of child terms that are connected to this parent term. And then this way you can avoid things like typos or having to, search for the exact wording of things. It's already there for the researcher to then plug in and go. And then finally you have your sync, so the data or the data file that comes out of this. So here's an example. If I'm running, a mass spec experiment, I have my instrument model as a parameter. And here the drop-down menu would give me the option for the instrument. I can click it, highlight it down along the entire column, and then I can add in the exact instrument model that was used for this experiment. So the tool that we have developed in order to assist us with this is called Swate. So it's a simple Excel plugin at the moment. I believe it's being developed for other spreadsheet domains, but right now it's very easy to load as a plugin for Excel. And you can see here on the right-hand side, you can add your building blocks. So you can select whether something's a factor or a parameter, and then type in the name, or it'll give you options for names as you start to type in, and then you can add that column. So here's a bit of a closer look of that. I have my source and different characteristics. So the sample label, the temperature at which the sample was processed, and then my data file or sample, the last column there, the data file name is then where the information related to that original sample is located. And you can see here on the right-hand side, it's easy to add a new parameter. You can start to type in the word instrument, and you get a bunch of different options to select. And then once this is selected as a parent term within your column, you then are given options to annotate with child terms. So we've taken this one step further as well, because as simple as it sounds, once you sit down with your experiment in an empty spreadsheet, it can kind of be difficult sometimes to say, ooh, what do I actually need? So at DataPLANT, we are constantly developing and talking about different ways of translating checklists from different experiments into the relevant templates. So when you log on to DataPLANT or log into Swate, you have template options. So if you're doing an RNA-seq experiment versus a phenotyping experiment, we've already begun devising, and we're constantly updating our templates so that the researcher already has a place to begin. Of course, an important component of DataPLANT as well is we don't want to force the researcher to do anything. So you don't have to use a template if you don't want to. You can design your own and save it if you know that this is going to be useful for yourself or maybe your lab group in the future. And in this way, you can then easily refer back to it. But it's up to people if they already have a good idea of what needs to be included, or else maybe they have no idea what requirements there are for a particular experiment or something along those lines. So just a little bit about the behind the scenes of Swate and how this actually works. So Swate is connected via a swoop up service, which is constantly updating. Is connected to a database of ontologies. So we have a combination of external ontologies that are related to the plant sciences, as well as our own NFDI for plants ontology. And what these do is, as terms are included into these ontologies, and as people are working on developing metadata templates, this is constantly being updated via swoop up so that SWAIT is as updated and ready to go as possible for the researcher. So just a bit about ontologies and why they're so important to SWAIT and to the metadata annotation. So I think many people might think of ontologies just as a dictionary. So, you look into an ontology file, you see a word, you see the textual definition, and in a way it is that, but ontologies also bring structure and context to these terms. So you're able to use things like ISA relationships to link terms to each other. So, you have the instrument model of a mass spec. So we're actually able to build relationships between terms. So these terms do not exist in a vacuum. Another really important component of these ontologies is that every term has an ID number, which means that this label now makes this word machine readable. So this is not only useful, for people looking through spreadsheets, but then later you can search through different ARCs or different spreadsheets to find specific terms that you are needing. So in addition to the external ontologies, I want to talk a bit more about our specific NFDI for plants ontology. So we have developed this in order to help sort of bridge the ontology gap. And what I mean by this is often these large, well-established ontologies, it might take a variety of different forms or quite a bit of time and effort to add a new term into an ontology. And the reality is, even though it doesn't feel like it after analyzing 1,000 samples, science is always changing and always updating, even though we feel like sometimes we're doing the same thing over and over again. So this ontology helps the researcher, if they go to type in a word or a new term that doesn't exist in one of our included external ontologies, they can easily, fill out a simple form that then gets forwarded to the DataPLANT team that we can then add to our broker ontology. And so that means that that term is then available to the researcher as soon as possible. We then plan to, by this little double arrow here, over time, we will work to push that particular term or word to the relevant external ontology. I don't want to get too into ontologies, but each one covers a specific domain. And we imagine that many people from all different areas of the plant sciences will be suggesting words for our ontology. And so it makes no sense for it to just sit there. It instead will eventually hopefully be pushed to the relevant external ontology, and that way it can be incorporated by the wider research community. But again, that responsibility is not on the individual researcher who just maybe needs it in that moment to annotate their metadata. It'll be instead on the responsibility of DataPLANT to follow through with this and make sure that term is then available. So as I've written down here, any participation is welcome. So we're always looking for new terms or new relations that might need to be built. So definitely check that out if you're interested. So yeah, so combination of these ontologies as well as these easily customizable templates, you're able to realize lab-specific metadata requirements. So whether you're doing a simple measurement or a very complex analysis, there's an easy way to keep track of all the metadata surrounding that. In addition to, say, an RNA sequencing or a DNA sequencing experiment, we also are developing templates specifically for repositories. So what I mean by this is if going into an experiment, you are going to upload your data to metabolites. You already know that that's a possibility. Then you can select a template that already has that repository in mind. So it already uses the terminology in that repository so that when you go to upload your metadata or upload your data at a later point in time, you don't have to sit there for hours, sifting through, trying to, translate your terms into what metabolites requires. We've already developed templates that in theory do this for you. This means that there's some really cool advantages of ARCs. And the first being that you can easily, share and work together with other people. So once you upload your ARC to GitLab, Other people with profiles are easily able to access this ARC, and in this way, things can be shared seamlessly between researchers. So whether that's the person sitting, down the hall from you, or the person halfway across Germany that needs a bit of your data, they're easily able to access this. If you'd like to maybe take a look at what some ARCs look like, we do have a number of ARCs on our registry. So here are some simple examples. We are working on making ARCs more complex, because I think most of us know a RNA-seq run is not going to get a journal published. So you need, other data with that. And in that sense, ARCs can also combine different types of data. But we're also developing those at the moment. Another thing I'd like to point out is this globe here. This means that the ARC is open to the public. So this is available for anyone to click on and look through. Of course, this is not a must. I think we often do not want to make our data readily available until it's ready for that. So in that sense, you can put different privacy settings on your ARC, either for just yourself. Maybe you just need to take some simple, plant height measurements in lab, doesn't relate to anyone else. You can make that private for yourself, or maybe just open it to the other collaborators within your group. Because ARCs have this standardized structure, so the assays, the runs, studies, workflows, once you understand this concept, you can easily look into any ARCs in order to find the information you need. So this allows for collaboration even across different research disciplines, because things should theoretically be in the same place no matter what the discipline is, so you can easily look up the data that you need. Because we upload our ARCs, or because our ARCs can be uploaded to GitLab, you're able to track changes, and previous versions are always available. So if there is something accidentally deleted that you need at a later time, you can easily go back in and restore this. You're also able via GitLab to contribute to multiple ARCs. So you're not limited to just ARCs that you've created, but you can also contribute meaningfully to ARCs from your research colleagues. This is also then kept track of on your profile, and people can see where exactly your contributions are. So that's handy if, I don't know, you forget what data you've uploaded or added somewhere. You can easily look it up. I think another very useful thing about ARCs is that information can be inherited from one to another. So this touches back on, I think, my second slide, where you might have collected data that was not important or relevant at the time, but maybe the following year you realize it's very handy to have for another experiment. So you can easily take that subset of data and transfer it to your current ARC and then work on it from there. So in that way, your data becomes reusable, because you can easily find it and move it to where it's now currently useful. We are also working on getting DOI numbers for ARCs. We are working on this so that ARCs can eventually be published or easily referenced in manuscripts. So this way you can, pack the data that's relevant to the publication, and people can easily click on that link and find your ARC without having to search too much. We are also working on converters. So in addition to our SWAIT templates, which might keep a repository in mind, we also are working on converters that can then, convert your particular templates into the repository friendly format. So, maybe at the start of your experiment, you didn't think you'd be uploading something to a repository, or maybe halfway through the experiment you realize, metabolites might not be the best target for my data. I'll instead upload somewhere else. So in this way, converters can actually map particular terms or data to whatever you may require later down the line. So here's just another quick outline of the whole DataPLANT metadata toolchain. So you can see here, a central component of this is the ARC, which can then be, as I mentioned, uploaded to GitLab, or else with the help of our converters, converted for a repository at the end of the experiment, or before journal publication. To help build an ARC or, to get you started, we have the ARC Commander tool. So this is an easy way of building, maintaining, as well as sharing ARCs. It gives you sort of a, step-by-step instructions and I believe a command line executable script. I think we are also working on a user interface for this. So for people who are not big fans of the command line, we're working on a user-friendly user interface, which is a bit easier to navigate. So again, you don't have to use this if you already have your folder, you know how to structure things, but this is just a quick, easy way of getting the structure you need, as well as the relevant, the beginning spreadsheets to annotate the investigation metadata. And then as part of the ARCs, we have our SWAIT tool, which I talked about, and how the different external and DataPLANT ontologies are linked to SWAIT, as well as the metadata templates. So in addition to these tools, we also have humans that can help with your data annotation and organization. So we have technical digital assistants, so people who, if you're having an issue with a particular tool that's not working, or you really need assistance in some way, we do have this digital assistance. And we're also working on the personal side of this as well. So what I mean by this is data stewards who will then help you maybe in person, but also, structuring your data. If there are, if you do have a very large experiment from all sorts of different discipline, RNA sequencing plus phenotyping, that they can help you combine and structure this according to FAIR principles. So not only assistance on the tool side, but also on how to best structure your data. So in addition to these various tools, as well as the community of experts that we are currently building, we have some other additional tools. So for example, we have our data management plant generator, and this can help you create a data management plan in a few steps. So it's something it's good for, or I think it's developed in mind for DFG projects as well. If you just need to summarize funding proposals, there's easy ways that you can create these plans in a few simple steps. So that saves time as well as effort. The night before a proposal is due, you realize you forgot to make this management plan. This is a quick and easy way of doing this. In addition to myself, feel free to ask me questions, but if there are questions or things down the line that you'd like to have answered, or if you're looking to, make contact again, we have some handy links here. So our help desk, as well as a general email address, and then of course our NFDI for Plants website. So feel free to check those out. And again, we're at the point where the more participation and contribution, the better, so we can get things really built up. So with that, I'd just like to thank the NFDI and the DFG, as well as the whole DataPLANT team. As you can see, we are all over Germany, and we're constantly building our network and, trying to get things really, built up. So with that, thank you for your attention, and open for any questions.